slow process. The next, enzymes do not alter the equilibrium constant of a reaction, but alter the rate of equilibrium is reached. The enzymes are very specific, and their specificity is retract to the substrate and the reaction they catalyze. So these are the characteristics of the enzymes. The main characteristics: enzymes are catalysts. The mostly enzymes are proteinaceous in nature, with some exception of all enzymes are proteins, but all the proteins are not enzymes. All the enzymes are proteins, but all the proteins are not enzymes. So these enzymatic proteins consist of mainly 20 types of amino acids. The polypeptide chain or chains of an enzyme, it is so tertiary in structure. The enzyme's tertiary structure, it is a very specific and important for their biological activity. Some enzymes like pepsin, amylase, urease, so these are made up of protein, like simple proteins. But most of the other enzymes have a protein and a non-protein component, both of which are essential for enzyme activity. The protein component of enzymes is known as apoenzyme, whereas non-protein component is called cofactor or prosthetic group. It means in each enzyme, it has a specific groups. One is the prosthetic group and a non-prosthetic group. Protein component that is called as apoenzyme, it is a protein component, and a non-protein component that is called cofactor or prosthetic group. The apoenzyme and prosthetic group together form a complete enzyme that is called holoenzyme. See, so this is holoenzyme. What is the holoenzyme? They are given chemical nature of enzyme. J.B. Summer explained that proteins explained that enzymes are protein in nature, and many enzymes require the presence of other compounds. Many enzymes require the presence of other compounds such as cofactors, coenzymes, and metal ions for their catalytic activity. So this entire active complex is referred as holoenzyme. 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 It is a combination of apoenzyme plus cofactor is equal to holoenzyme. The entire active complex. Active complex. What is the active complex? It is a combination of apoenzyme plus cofactor. So that is called. So this is active complex. So this active complex is referred as holoenzyme. So this activity of enzyme is due to cofactor So this is follow enzyme. Next, specificity of enzymes. Specificity of enzymes. What is the specificity? Specificity. It is one of the property of enzyme, that is specificity. Specificity means enzymes are reactions in specific. Enzymes are reactions in specific. One enzyme catalyzes only one specific reaction. One enzyme catalyzes only one specific reaction. So these are acts on a particular substrate. Acts on a particular substrate. For example, enzyme sucrase catalyzes the hydrolysis of only sucrose and no other substrate. It means enzymes reactions are in specific. One enzyme catalyzes only one specific reaction. It means specificity. It is one of the important property of enzymes. The main important properties of enzymes, one is the catalytic property, and the next one specificity, and reversibility, and thermolability, sensitivity of pH, and proteinaceous in nature. These are main properties of enzymes. In that, one of the property that is given specificity of enzymes. So one of the properties of enzymes, so it is one type of property of enzyme, that is specificity. In that specificity, different type of specificities are given. One is the absolute specificity. Absolute specificity. Absolute specificity, it means the enzymes will catalyze only one reaction by acting on a single substrate. Single type of substrate. That is absolute specificity. The example that is given, glucokinase, which converts only glucose to glucose 6-phosphate. So this is glucose kinase enzyme. In this reaction, glucose convert into glucose 6-phosphate in the presence of enzyme glucokinase. In this reaction, one of the phosphate molecule which is utilized, it means ATP convert into ADP. It means one phosphate which is utilized in this reaction. Glucose convert into glucose 6-phosphate in the presence of glucose kinase. So this is absolute specificity. Glucose kinase converts into glucose 6-phosphate. And second one, group specificity. First one is absolute specificity. Next, group specificity. In this group specificity, the enzyme will act on a group of related molecules. Enzyme will act on a group of related molecules. As in first case, enzyme which catalyzes only one reaction on a single type of substrate, but in group specificity, enzymes will act on a group of related molecules. For example, they are given a hexose kinase which acts on all hexoses and converts to their hexose phosphates. Hexokinase, you see hexose sugar. So these hexose sugar convert into hexose phosphates. Hexose phosphates. One is the glycogen starch and pentose phosphate pathway and the glycolysis pathway. So this during this glycolysis pathway, energy which is liberated. So this group specificity in this type, hexose kinase, it is a one type of enzyme. So these hexose kinase acts on the all hexose phosphates. So these are the hexose phosphates. So this type of specificity is called group specificity. So specificity, it means enzymes catalyzes one specific reaction or acts on a particular substrate, that is called specificity. And the third one, linkage specificity. Linkage specificity. In this type of specificity, enzymes will act on a particular type of chemical bond. Particular type of chemical bond. Proteases acting on various proteins. Proteases act on various proteins. This is a best example of linkage specificity. And the fourth one, stereochemical specificity. Stereochemical specificity. Enzymes will act on a particular optical isomer. This image L amino acid oxidases. L amino acids acting on only L amino acids. So this is L amino acid oxidases. So these are react on only L amino acids. It is a stereochemical specificity. Now the next one,
Sir, you are in mute. Sir, your voice is not audible, sir.
wait, 1 second. So it is clear? Hello?
Yes, sir.
Okay, ma.
Yes, sir.
Now the next one, see in this image, it is enzyme. In this enzyme,
Substrate.
so this portion, this is called active site, and it is a substrate. The substrate and the active site of the enzyme. Substrate which is act on the active site of enzyme. Now the formation of enzyme substrate complex. Enzyme substrate complex. So what is the enzyme substrate complex? Let us see. So active site, so this is active site. It is a, what they are given, active site, it is a part of an enzyme. So it is enzyme. In this enzyme, it is a small part that is called active site. So what is the function of active site? Where the substrate bind and undergo a chemical reaction. Substrate which is present, so these substrate bind specific site in the enzyme. So this specific site is known as active site. Active site, and this enzyme and a substrate together is called enzyme substrate complex. So the active site of an enzyme is usually found in a cleft or packet. So this is a packet that is lined by amino acid residues. Enzyme substrate complex. And this enzyme substrate complex next classification of enzymes. Classification. So these enzymes are classified into different types. Before going to classification, one of the most important, that is naming of an enzymes. Naming. How to naming the enzymes. One second. Naming. Naming of an enzymes. Naming of an enzymes. So naming of an enzymes. How to given the name of enzymes. So these naming of enzymes can be done by three ways. First way, substrate acted upon by an enzyme. substrate acted upon by an enzyme. You see in this image substrate and it is enzyme. So first one, naming, how to naming of an enzyme. First rule is substrate acted upon by an enzymes. For example, what are the examples? Substrate acted upon by an enzyme. First one, urease. Urease acts on urea. Urease acts on urea. And second one, cellulase. Cellulase, it is acts on cellulose. And third one, sucrase. Sucrase, that is acts on sucrose. Substrate acted upon by enzyme. Urease acts on urea. Urease, it is enzyme. So what is the substrate? Urea is a substrate. The next, cellulase, it is a enzyme. It is acts on the substrate, that is cellulose. The next, sucrase. Sucrase, it is a enzyme that is acted upon by which substrate? That is called sucrase. Next, maltase. Maltase, very good. Maltase, Maltase. Maltase, it is act on maltose. Next, nuclease. Nuclease. Nuclease, it is acts on, what are the nuclease? It is acts on DNA or RNA. DNA or RNA. Okay. Nucleases are acts on the DNA or RNA. And lipase. Lipase. Lipase, it is acts on lipids. Okay. So it is naming of an enzymes. First case, substrate acted upon by an enzymes, that is urease, urea, cellulose, sucrase, sucrose, maltase, maltose, and nucleases acts on nucleic acids such as DNA or RNA, and lipases acts on the lipids. Now the second case, type of reaction catalyzed by an enzyme. Type of reaction catalyzed by an enzyme. Type of reaction that is catalyzed by an enzyme. For example, carboxylases. Carboxylase help in carboxylation process. Carboxylases help in the carboxylation process. Next one, dehydrogenases. Dehydrogenases. So these dehydrogenases help in the dehydrogenation. Dehydrogenases help in the dehydrogenation. Next one, oxidases. Oxidases help in the oxidation. Oxidation. Phosphorylases. Phosphorylases help in the phosphorylation process. Phosphorylation process. It is the second rule by the naming of an enzyme. First one, substrate acted upon enzyme. Second one, type of reaction catalyzed by an enzyme. This is second rule and this is the first rule. Okay, substrate acted upon by an enzyme. Second one, type of reaction. What is the reaction? As in carboxylase case, carboxylation. Second one, dehydrogenation. Oxidases, that is oxidation. The phosphorylases, that is phosphorylation. And next, third one, a combination of both substrate and the type of the reaction. A combination of both. What are the both substrate and the reaction, and the type of reaction. You see, in the first case only substrate. Now the second case only type of reaction. As in third case, combination. It means substrate and type of the reaction. These both are involved in the third case. First one, for example, malate dehydrogenase. Malate example, malate dehydrogenase. Malate dehydrogenase, it is an enzyme. It is helps in malate dehydrogenation. Helps in malate dehydrogenation. Okay, malate dehydrogenation. Second one, citrate synthetase. Citrate synthetase, it is an enzyme, helps in the citrate synthesis. Citrate synthesis. And third one, pyruvate kinase. Pyruvate kinase. These pyruvate kinase helps in the pyruvate synthesis. Pyruvate synthesis. So this is naming of an enzyme. One is the substrate acted upon enzyme. Second one, type of reactions catalyzed by an enzyme. Next one, combination of both substrate and the type of reaction. Next, classification of an enzyme. So the International Union of Biochemistry, International Union of Biochemistry classified enzymes. International Union of Biochemistry IUB, it is classified and enzymes. Classifieds and enzymes. One second. So in the year of 1964, the International Union of Biochemistry classified enzymes into main 6 major classes. They are oxidoreductases, transferases, hydrolases, liases, isomerases, and ligases. The year of 1964, International Union of Biochemistry, entire the enzymes which are classified into 6 categories. They are namely oxidoreductases, transferases, hydrolases, liases, isomerases, and ligases. This first class, that is oxidoreductases. Oxidoreductases. So these oxidoreductases, these enzymes catalyzes oxidation and reduction of substrates. Oxidation and reduction of substrates, usually involving hydrogen transfer. Hydrogen transfer. Oxidoreductases catalyzes the oxidation and the reduction reaction, where the electrons transfers takes place from one form of a molecule to other molecule. See this, and example that is given, pyruvate dehydrogenase. Pyruvate dehydrogenase. You see in this reaction, as in first case, the hydrogen, what the definition they are given?
Transfer of
hydrogens from one place to another place. Hydrogens or electrons. You see, these hydrogens transfer from one place to another place. As in first case, A. Okay, AH2. So these hydrogens transfer to B. So this is oxidoreductases. So these oxidoreductases again divided into 3 subclasses. Oxidoreductases again divided into 3 subclasses. That one is the dehydrogenases, second one reductases. Second. Right. Oxidoreductases. Oxidoreductases. It is the first class oxidoreductases. In these oxidoreductases, transfer of hydrogens or electrons from one place to another place. So these oxidoreductases further classified, divided into how many types? 3 types. Okay. First one, dehydrogenases. Dehydrogenases. So dehydrogenases. So these dehydrogenases transfer hydrogen atom from the substrate to NAD plus. Transfer the hydrogen atom from substrate to NAD plus. Okay. Dehydrogenase, it means transfer hydrogen atom
From
substrate to NAD plus. For example, you see example of the dehydrogenases. Malate combines with NAD plus and gives rise to see like this, rise to okay, gives rise to oxaloacetate plus NADH plus H plus. Okay. So this is the example of dehydrogenases. Malate combines with NAD plus and forms oxaloacetate plus NADH plus H plus in the presence of an enzyme malate dehydrogenase. So malate dehydrogenase in the presence of enzyme malate dehydrogenase. Okay. Malate combines with NAD plus and forms oxaloacetate plus NADH plus H plus in the presence of enzyme malate dehydrogenase. So this is dehydrogenation, and the second one, reductases. So in this case, they add hydrogen to substrate. In this case, in reductases case, they add hydrogen. From the substrate. They add hydrogen to substrate or removing oxygen. Substrate or removing oxygen. As in first case, transfer hydrogen atom from substrate to NAD plus hydrogen atom substrate. So this is substrate to NAD plus. Now they can form oxaloacetate plus NADH plus H plus. As in the second case, reductases, they add hydrogen to substrate, add hydrogen or removing oxygen, addition or deletion, addition of hydrogen or removing of oxygen to the substrate, that is called reductases. For example, example nitrite forms nitrate. Nitrite forms the nitrate, NO3 minus. So as in case NO2 minus, nitrite. Nitrate. In this reaction, the enzyme which is involved, nitrate reductase. Okay, so this is reductases. Now the third one, oxidases. So in this case, transfer of hydrogen from substrate to oxygen. Transfer of hydrogen
and subtract to
from substrate to oxygen. Substrate to oxygen. Okay, transfer of hydrogen from substrate to oxygen. First case, transfer of hydrogen substrate to NAD plus. As in third case, hydrogen from substrate to oxygen. So this is dehydrogenase and this is oxidases.
Sir, once
This
again
case,
repeat, no. Excuse me, sir. Once again repeat, no.
okay, ma.
Morning నుంచి ఈవినింగ్ వరకు ఇవి క్లాసెస్ ఓన్లీ సండే సాటర్డేస్ ఓన్లీ సండేస్
Please mute your phones. So oxidoreductases,
ఫస్ట్ సాటర్డే
these are three types. So these oxidoreductases again divided into three types. They are first one dehydrogenases, second one reductases, and the third one oxidases. In the first case, dehydrogenases, hydrogen atoms transfer from substrate to NAD plus. Hydrogen atoms transfer substrate to NAD plus.
Straight to
NAD plus. You see, in the third case, oxidases, hydrogen atoms transfer substrate to oxygen. Okay,
transfer of hydrogen from substrate.
okay,
Same.
both are not same. As in first case, hydrogen transfer
NAD.
NAD plus.
Substrate to NAD plus.
As in third case, hydrogen transfer substrate to oxygen, not
Oh,
NAD plus.
First NAD plus third
Third
oxygen.
one oxygen. Okay.
Okay. Okay.
So example, glycolic acid. Glycolic acid combines with oxygen. Combines with oxygen, gives rise to glycolic acid. Glycolic acid and
Yes.
forms H2O2. Okay. You see,
Oh.
glycolic acid,
H2.
hydrogens substrate to oxygen. Okay, so these glycolic acid. In the presence of enzyme glycolic acid oxidase. Glycolic acid oxidase. Okay, glycolic acid oxidase. So these are the oxidoreductases. Three types, 1, 2, and 3. Now the next one, second one, transferases. Transferases. So these transferases, so these enzymes are involved in the transfer of a chemical group. Transfer of a chemical group. So what is the chemical group? Such as amino acid, amino group or phosphate group. Transfer of a chemical group such as amino group or phosphate group from one molecule to another. Okay, amino group or phosphate group from one molecule to another. Transferases. So these transferases are catalyses, transfer the chemical group. What is the chemical group? That is amino group
Amino
or
group or phosphate group.
phosphate group from one compound to another compound. So that is called transferases. So transaminase, it is a best example of transferases. So this transaminase transfers an amino group from one molecule to another. Okay, one molecule to another. For example, aspartic acid combines with alpha-ketoglutarate acid. Aspartic acid is a amino group. Okay, because aspartic acid, that is one type of a amino acid. For example, so this is aspartic acid. Aspartic acid combines with alpha-ketoglutarate acid. So these are gives rise to oxaloacetic acid plus glutamic acid. Okay, transaminase transfer of one group to another group.
Group 2.
Sir, page number, please.
So these are classification page number
16.
I don't know
16.
page number direct.
No, it's not there.
Next,
Just table ఒకటి సార్.
దీనిలో ఓన్లీ టేబుల్ ఉంది సార్.
transfer oxidoreductases, transferases, and the third one hydrolases.
సార్ ఇది ఇంపార్టెంట్ సార్ ఎగ్జామినేషన్ పాయింట్ ఆఫ్ వ్యూలో.
ఇంపార్టెంట్ అన్న, ఇవి క్లాసిఫికేషన్ కదా. ఇచ్చింది ఆల్రెడీ నామినిక్లేచర్ అండ్ క్లాసిఫికేషన్ ఆఫ్ ఎంజైమ్స్ ఉంది కదా.
లేదు సార్.
ఉంది పేజ్ నెంబరు
ఇచ్చిండి సార్.
ఇచ్చిందమ్మా మీరు ఎంజైమ్స్ లో 2.3 ఒక టేబుల్లో ఇచ్చారు చూడండి. ఆక్సిడేటేజెస్, ట్రాన్స్ఫరేజెస్, హైడ్రోలేజెస్, లైయేజెస్, ఐసోమరేజెస్ అండ్ లైగేజెస్ 2.
ఈ టేబుల్ చాలా సార్.
6 types of songs.
క్లాసిఫికేషన్ ఇచ్చాడుగా.
Page number 66.
పేజీ నెంబరు తెలియదు నాకు, చూడండి మీరు.
160 sir.
ఓకే. నెక్స్ట్ ఫస్ట్ కేస్ oxidoreductases and second one transferases and third one hydrolases. So these hydrolases, so these group of enzymes involved in the cleavage of bonds for the addition of water, cleavage of bonds by the addition of water, hydrolases. So they are hydrolytic enzymes which catalyzes the hydrolysis reaction by using of water molecules to cleave the bond and hydrolyze it. The clear definition that is given, hydrolases.
Yes, sir.
So they catalyze the hydrolysis of a bond. Example, pepsin hydrolyzes peptide bonds in proteins. Okay, hydrolases. It means these hydrolases enzymes involved in the cleavage of bonds by the addition of water, that is called hydrolases. Cleavage of bonds by the addition of water. Now the next one, liases. So these are also enzymes involved in the removal of phosphate from the substrate. Liases, removal of phosphate from the substrate. See, they are enzymes that catalyze body work, breaking a chemical bond through not involving hydrolysis or oxidation and forming a double bond or adding a group to double bond. So liases is nothing but splits the substrate in the absence of H2O. Split the substrate, absence of H2O. Isomerases. So these enzymes are involved in the rearrangement of atoms within the molecule. Isomerases, they are a class of enzymes that convert a molecule from one isomer to another isomer. Isomerases facilitate intramolecular shift arrangement. Okay.
So in the lysis, how we have to know how to write the definition or an example?
Liases are the enzymes split the substrates in the absence of H2O, absence of water. Right, that is H2O. Liases.
So here in the reader 6.1,
hello, ask me now. What is your doubt?
Sir, in the lysis 6.1 in the reader page number 16, C minus O and C minus S, C minus N and C and C. So how we have to write these examples in the exam or in the
Liases.
Yeah, liases.
So that is enzyme liases.
Ligases. Excuse me, it is ligases. Yeah, ligases.
In the second box,
in the PC lysis,
catalyze the cleavage of carbon bonds. In the first case, CC bonds, it means carbon bonds Now the second case carbon oxygen. Okay.
Carbon, oxygen. Okay.
And third one carbon nitrogen. Okay. So these bonds cleavage the carbon carbon bonds and certain other bonds. Okay. Already they are given clearly. Breaking a chemical bond. You see what they are given, breaking a chemical bond. What is this chemical bond? Like CC bond or CN bond or CO bond. Okay.
Okay.
Through the not involving hydrolysis. Breaking a chemical bond through not involving hydrolysis.
So in the reader, it has given that catalyze the cleavage of carbon, carbon atom, and the certain other bonds.
Other bonds.
And
What
the-
are the other bonds? Such as C double bond O, carbon oxygen or carbon nitrogen bond.
Nitrogen bond. Okay.
That is liases.
Carbon and sir, C is sulfur. Carbon is
అది లైగేజెస్ లో సల్ఫర్. ఇక్కడ రెండు ఉన్నాయి, లైయేజెస్ వేరే, లైగేజెస్ వేరే. లైగేజెస్ అంటే అవి యాడింగ్.
Yeah.
ఓకేనా? లైయేజెస్ వేరే, లైగేజెస్ వేరే.
double bond is liases. Single bond is ligases.
Ligases, yeah.
So this liases and isomerases. Next ligases. You see ligase, ligases. Ligase catalyzes the ligation. Ligation, it means joining of two large molecules. Okay, joining of two large molecules by forming a new chemical bond. Forming a new chemical bond between them. Ligases. So these ligases are associated with the formation of new bonds. Ligases are associated, you see, A plus B gives rise to AB. AB, it is a new compound. A and B, these are separated. It means A combines with B and forming AB. Okay.
So carbon plus oxygen give rise to carbon dioxide, like this.
Like that.
For example.
For example, ligases. See, glutamic acid. Glutamic acid combines with NH3.
NH3.
Glutamic acid plus NH3. One second.
Okay. Glutamic acid plus NH3.
Glutamic acid combines with NH3 plus ATP. ATP gives rise to glutamine. Glutamine plus ADP. You see, in the presence of enzyme, glutamine synthetase. Glutamine synthesis okay. So this is the
now glutamine synthesis is the ligase, sir.
Ligases. Glutamine synthesis, it is an enzyme. So these enzyme, what is the glutamine synthetase? It is a combination of two compounds, that is glutamic acid plus ammonia. You see, so this is glutamic acid and ammonia. So these two compounds are formed, combines and forms a new compound that is called glutamine. Okay, glutamine. In this reaction, one phosphate molecule that is utilized. You see, ATP. ATP, the next formation of ADP.
ADP.
This one phosphate molecule is utilized in this reaction. Glutamic acid plus ammonia in the presence of ATP and forms glutamine and EADP plus EADP.
Excuse me, sir.
Glutamine
Here,
synthetase is a
glutamine
enzyme.
synthesis is a ligase.
Ligase enzyme. It is a enzyme, ligase enzyme.
Belongs to
Okay.
ligases.
Glutamine synthetase. Okay, so this is the classification of an enzymes. The enzymes are mainly classified into broadly 3 types. Okay.
According to solubility also they are classified now, sir.
Yes, ma. Next.
So one doubt, sir.
Uh,
In the example classification of enzymes means you have to write this oxidation reductase,
right.
transferase, hydro. This all. Okay.
Classification
Definition
of enzymes.
and the example. That's all.
Right.
Okay. This answer will come essay or short?
Short question.
Short question.
Hmm.
Okay, fine. Sir.
Next. Enzyme cofactor. Enzyme cofactor. Cofactors. Cofactors are non-protein chemical compounds. Non-protein chemical compounds. Already in first or second slide, we are clearly given what is the non-protein part and what is the protein part. Non-protein component is called cofactor. Okay. So these cofactors are also called prosthetic group. You see. So this is a non-protein part. Okay, na? Non-protein part is known as cofactor or prosthetic group. Now, what this is enzyme cofactors. Cofactors are non-protein chemical compounds. So these are associated with enzymes. So,
Sir, can you send notes, please?
okay.
Excuse me, sir, can you send notes?
Okay, Nana. So these cofactor acts as a catalyst and regulates the functioning of an enzyme. So holo enzyme. So this is apo enzyme and combination of cofactor and they forms holo enzyme. In this holo enzyme, so it is an enzyme and it is a active site. In this active site, so this green color portion that is called as substrate, red color enzyme, and this empty portion, so this portion is called active site. On this active site, the substrate binds on the enzyme at a specific site that is called active site. Next, cofactors. There are 3 kinds of cofactors present in enzymes. What are the 3 cofactors? It is important. Prosthetic groups, coenzymes, and metal ions. What are the components are present in cofactor? Is a important question. One is the prosthetic group. Prosthetic group is nothing but it is a non-protein part. Okay, and it is also called as cofactor. So these prosthetic groups are covalently or permanently bound to an enzyme. Prosthetic groups permanently bound to an enzyme at all times. Okay, prosthetic group. So these prosthetic groups are organic compounds. They are formed from other cofactors.
Example.
Second one, coenzyme.
NAD.
These
For
coenzymes,
example, for prosthetic groups, sir?
prosthetic groups,
Yes, sir.
FAD.
peroxisomal enzymes. Prosthetic group, for example, you write that peroxidase, the catalase, peroxidase and catalase. These are the prosthetic groups which catalyzes hydrogen peroxide to water and oxygen.
Yes, water and oxygen.
Oxygen. That is the prosthetic group. Second one, coenzymes. What are the coenzymes? Prosthetic groups are organic compounds and non-protein parts. And coenzymes, these are also organic and non-protein compounds. And these are binds to enzyme only during catalysis. So these coenzymes first discovered by Fritz Lipmann. Coenzymes first discovered by the scientist name of Fritz, F-R-I-T-Z, Fritz Lipmann, Lipmann. Fritz Lipmann first discovered these coenzymes. And these coenzymes are not only assist enzymes in the cleavage of the substrate, examples NADP, FAD, these are the coenzymes. NADP, it means nicotinamide adenine dinucleotide phosphate, NADP. And FAD, flavin adenine dinucleotide. And NAD, nicotinamide adenine dinucleotide. NADP, and FAD. These are coenzymes. And third one, metal ions. Metal ion
is nothing but inorganic ions.
Organic
Sir,
ions
excuse me.
like
Can you
zinc, magnesium.
Excuse me, sir.
Okay.
NAD. What about NAD?
Nicotinamide dinucleotide.
Nicotinamide
Dinucleotide. Metal ions in some enzymes having metal ions. Such enzymes are called metalloenzymes.
In the reader, it has given inorganic ions. So that's I asked you, that is nothing but metal ions.
Metal ions.
Is the same?
Yes.
One and the same. Metal ions, or we can write inorganic ions.
Inorganic ions, very good. Same.
Okay.
Both are same. So where the enzymes having metal ions, such enzymes are called as metalloenzymes. Some are the Examples of metalloenzymes. One is the cytochrome
Iron.
oxidase. Cytochrome oxidase. Oxidase is a metalloenzyme. And hexokinase. Hexokinase. And catalase, nitrate reductase, and carbonic anhydrase. These are the metalloenzymes. So in these enzymes, metal ions such as iron, magnesium, manganese, molybdenum, and zinc. So these are the metal ions which are present in metalloenzymes. Okay, these are the cofactors, kinds of cofactors present in the enzyme. Next, mechanism of enzyme action. Mechanism of enzyme action. So what is the mechanism? So the energy is required to bring the insert molecules into the activated state. If the molecules is inserted, some of the energy is required. So the amount of energy required to raise the energy of molecules at which chemical reaction can occur is called activation energy. During the reaction, some of the energy is required. Such energy is called activation energy. Okay. So in mechanism
Example,
of,
amino acid.
wait, activation of energy. If the activation of energy is required for the formation of enzyme substrate complex, so the molecules can be participate in the reaction. Suppose mechanism of enzyme reaction, you see, so this is an enzyme and it is a substrate. It is a reversible reaction. Okay.
Yes.
So this enzyme combines with the substrate and the formation of enzyme substrate complex. Okay. So during this reaction, some of the energy is required. Okay. Such energy is called activation energy, where the enzyme substrate complex is formation by the enzyme plus substrate. This reaction, some of the energy is needed. Such energy is called activation energy. Okay. Now, how these mechanism, how the enzyme mechanism, this mechanism of enzyme action, which explains by mainly two theories. One is the induced fit model and the second one lock and key model. Okay. Entire the mechanism of enzyme action, which is explained by mainly two models. One is the induced fit model.
Fit model.
Second one, lock and key model. You see, as in first case, induced fit model. So, as in first case, they are given 1, 2, 3, 4. First one, you see, so this is substrate, active site. So this portion is called active site, where the substrate binds to an enzyme at a specific site, that is called active site. Now, as in first case, substrate enters active site of enzyme. So this is called substrate, and this is called enzyme. So this portion is called active site. So where the substrate enters to active site of enzyme, so these are a mark. Now you see, as in second case, enzyme substrate complex is formed when the substrate enters on active site. Now the formation of enzyme substrate complex. So the enzyme changes shape slightly as substrate binds. So previously, so this is enzyme shape. After the binding of substrate, now the enzyme changes their shape. Okay. As in third case, substrate. So what are the substrates? So this is substrate. Substrate is converted to products. You see, as in first case, substrate, the color is given in green color. Okay, na? And dark color, it is indicates this is an enzyme. As in the second case, enzyme and substrate both are fused. Okay.
Fused.
Now this is called enzyme substrate complex. As in third case, the products are formed. Now the colors are changed. It means substrate is converted to products. Please mute your phone. Phone mute lo pettanamma. Next, fourth case. As in fourth case, products leave the active site of the enzyme. After the formation of the products, these are leave the active site. So this is induced fit model. Now the second one, lock and key. So this is key. Key stands for substrate, and lock, it is enzyme. Okay. You see, as in this image, the lock and key model was suggested by Emil Fischer in the year of 1894 and is hence known as Fischer's theory to describe the enzyme substrate interaction. Enzyme substrate interaction. So what is the enzyme substrate? According to Locke and theory model, active site of the enzymes. What is the active site? So this is active site where the key enters into the enzyme. Such place is called active site. Now the active site of the enzyme serves as the lock, while its substrate serves as the key. So this is key stands for substrate and lock stands for, so that is enzyme. Okay. So on that, the shape of active site of the enzyme is complementary to the shape of the substrate. The active site of enzyme can hold the substrate closer to the enzyme by forming an unusable intermediate compound, which is the enzyme substrate complex. Enzyme substrate complex. So according to this lock and key hypothesis, enzyme and its substrate have a complementary shape, and the specific substrate molecules are bound to a specific site of the enzyme molecule. So that is lock and key mechanism. It is proposed by the scientist name of Emil Fischer in the year of 1894. So this is mechanism of enzyme action.
Hello.
So overall, sir.
Hello. Sorry.
అయిపోయింది క్లాస్.
Information.
Is over.
No information.
Over.
Sorry.
క్లాస్ అయిపోయిందా? ఆ?
4:30 అయింది కదా, క్లాస్ ఈజ్ ఓవర్.
Class is over.
Okay. So
Okay,
the
bye
half.
friends.
Bye, friends.
Bye.
Hello.
Hello, madam. హలో మేడం, ప్రాక్టికల్ ఎగ్జామ్స్ ఉంటాయండి. హలో మేడం.
అందరు స్టూడెంట్స్. అందరు స్టూడెంట్స్